Thank you, and a special thanks to Larry. We actually coordinated our talks because he had all the introductory slides, so I can go more, and I warn you in advance, more into kind of the science of what is behind something like cell-based meat or cultured meat. So the challenge is for 2050 that we need to feed 10 billion people with high quality and sufficient food. The good news is that can be done. The bad news is not if we continue business as usual. But before I go into the cultured meat story, I want to reiterate a little bit of what Larry said about that food is essentially a bag of chemicals, sugars, amino acids, vitamins, minerals, calories. Most of it comes from plants. And plants have this wonderful system of getting glucose, or making glucose by getting CO2 out of the air, sunlight, a little bit of nitrogen and phosphorus out of the soil, and create glucose. And glucose is, in addition to a sweet kind of product, it's also the building block of most other components in our body, our fatty acids, our proteins. So, by using, again, nitrogen and phosphorus out of the soil, getting the sunlight and the CO2 and the glucose, and from that glucose you produce proteins, amino acids, fats, and other materials. It's a very, very essential part. The thing is that this is a function in a plant that is actually dependent on an invasive species. The chloroplasts, these little green things in the otherwise white plant cell, are invasive species. About between 800 million and 3 billion years ago, these parasites kind of invaded plants and started to become the energy factories of these plants. And that's pretty much, for a large part, what we survive on. Wonderful system, very, very efficient. You don't want to really mess with that too much. We can, and Larry already mentioned, we can actually synthesize all these components just by using the different atoms. This is a very boring slide, but this basically lists all the methods that have been used, and sometimes successfully, to create from the nitrogen, from carbon sources, CO2, from phosphorus and sulfur out of the soil, to create all the essential amino acids and sugars that we need to survive. It costs a lot of energy. It's way less efficient than a plant, right? But it can be done in case of emergency. The bigger problem in food, and it has been mentioned a couple of times, are the animals. We have been domesticating animals for the last, 10,000 years for our food, and it has become a really important part of our food system. And by the way, this is Hermione. This is a cow that was very famous in the Netherlands for a while because it escaped the slaughterhouse, and it went on a rogue life for about 3 months, evading everybody, and finally it got caught, and now it's in a shelter. And it really became very famous, which tells you the story that we kind of have sort of empathic relationship with these animals as well. We actually have in the Netherlands a program called cow hugging. You can do workshops in hugging cows. Whatever. We have a lot of cows. When I look at this animal, this cute animal, I basically look at a technology that converts vegetable proteins in its diet to animal proteins in our diet. And at that, and it was already referred to, it's very, very inefficient. It uses a lot of resources to get there. Having this animal, this technology as an intermediate in our food production system is really a drain of resources. If you nowadays would come up with a technology to convert plant proteins into animal proteins, you would never come up with a cow, right? It's that inefficient. All right. So, was already mentioned, and I'm going to skip over this very quickly. Greenhouse gas emission associated with cows. A lot of water used for 1 kilo of beef. We require 15,000 liters of fresh water. And I know you have plenty of fresh water in Finland, but there are areas on the globe where that's not the case. We need to think about these phosphorus and nitrogen cycles. Every time I see a really efficient food system, I start to think of, is this circular or not? Because you can create really efficient, actually a soybean is an extremely efficient food source, but it's not a sort of circular food source because most of it is used for animals. And already biodiversity was mentioned as well, and also at some point we may want to produce food at sort of remote places to reduce transportation, to improve accessibility either in remote places on Earth or in space. You already heard the quote of Winston Churchill, 1931. You may have been wondering how this guy actually came up with that quote. He was a friend of Alexis Carrel, a Nobel Prize-winning transplantation surgeon, who, together with Charles Lindbergh, the aviator, developed a system to keep organs alive outside of the body in 1930. And they succeeded. For a couple of weeks, months, they kept those organs alive outside of the body. For transplantation of organs, a very useful technology. And they noticed that these organs grew a little bit in size. And Churchill was a friend of Alexis Carrel and of Charles Lindbergh. And so, under the pleasure of cigars and brandy, they came up with the idea, oh, perhaps we can at some point use this for food. However, in 1931, the technology really was not there yet. It had to wait for the discovery of stem cells. Stem cells are cells in our body to regenerate our body. They're sitting there waiting to repair tissue when it's injured. The ultimate stem cell that we are all derived from is the fertilized egg, and that can make all sorts of tissues. And the embryonic stem cell, a lot of that has been discussed about that, also can make all sorts of tissues. What most people don't know is that almost all of our tissues in our body have stem cells. And they are a little bit more committed towards a specific tissue. And our muscle cells are really chock full of stem cells. Every muscle fiber has 4 or 5 stem cells, again, sitting there waiting to repair that muscle when it's injured. And when it's injured, they start to proliferate, and they start to repair that gap in our muscle. So if you tear your muscle, it's replaced by muscle tissue and not by scar tissue, which is a wonderful system. So the idea was very simple, referring to Winston Churchill, but now using the power of stem cell technology, is to poke a cow in the butt, get like 0.5 gram of muscle out of it. You can separate the fat tissue from the muscle tissue. Fat tissue also has stem cells. Fat, ladies and gentlemen, is a tissue. It's not just fat. It's a tissue. And amazingly, fat tissue also has stem cells to regenerate fat tissue, which we can all experience when we don't have enough of it. So we take that sample from the cow and we extract the stem cells. A small sample of 0.5 gram already has a couple of thousands of these stem cells. And we let them proliferate, because that's what they do very, very well. And this is basically the big sort of advantage of this system. The proliferation allows us to create much more tissue from that one sample, thereby reducing the number of cows on this planet, or any other animal, and reducing the environmental impact and the resource drain. Of course, cells, it was kind of referred to by Lowry as well in those berries. They don't taste really, they don't have the protein content that we typically want from them. So we want to make muscle tissue again out of that. And the first step is they need to merge into a muscle fiber. A muscle fiber is a multinucleated muscle cell. And that's a very simple instruction. We basically starve them, and then they start to merge, just like a plant starts to flower if you don't feed it. And then, of course, the muscle needs to perform labor, because we all know that labor actually beefs up our muscle, and that's really protein synthesis, if you think about it. And if you run a marathon, you get, you know, these people are really skinny, they don't have thick muscles. You have to go to the gym to get thick muscles. And why is that? It's not because the movement itself is a trigger for protein synthesis, it's mostly the tension that is the result of contraction. So pumping iron, lifting weights, gives you much thicker muscles, much more protein-rich muscles than running a marathon. So that's why we created this system. We have those small fibers, already primitive fibers, not a lot of protein yet. We put them in a circle around a central column. And amazingly, when you do that, these cells start to find each other. They start to attach to each other. They start to form a tissue. And what's more, they start to contract. Ladies and gentlemen, our muscle cells are exercise junkies. Our brains are not. But these are not connected to brains. Right? So they start to contract, and because they are in this ring structure, that contraction actually leads to tension, and that tension leads to protein synthesis. And after 3 weeks, we have a muscle fiber that under the microscope looks exactly the same as any muscle fiber that you take immediately from a cow. We created 10,000 of these muscle fibers. I can tell you it's really boring work. Really, really tedious. My technicians in the lab really hated me for about 3 months to make this. And we made a hamburger out of that. And in 2013, we did something very unusual. We presented this hamburger in a hybrid between a cooking show and a press conference for 200 international journalists in London. And the hamburger was cooked and eaten. I have a small snippet of the video here. We presented it in a Petri dish, which was kind of a gimmick to just show, oh yeah, this is cultured. By the way, it cost us €250,000 to make this hamburger. So it was not really a consumer product yet. It was eaten by 2 food journalists, Hanni Rützler from Austria and Josh Schoenwald from Chicago, and they said, oh yeah, this is meat. And they gobbled my €250,000 hamburger up in 5 minutes. This was not a product launch. It kind of felt like a product launch. And you may be wondering, 2013, 2019, 6 years, what the heck has this guy been doing? Why don't we see this in the restaurants and on our plates yet? And that's because there were a lot of problems still with the production method. Cell culture, if some of you know what that is, it's not easy to scale. It's not easy to make it efficient. We still needed animal products in this hamburger other than the cells. The cells always will be animal products. So we needed to work on that. And we needed to get rid of blood. When we culture cells, we typically use blood, at least 20%, to make them grow. When we put them in a ring structure, we use a gel that was still kind of animal-derived. And so we had to work on a lot of issues to eventually make this an animal-free product other than the cells, which is not a problem because they are replicative. All right, so I don't go into all the science because I don't have the time, but we spent 5-6 years on working through it. I just take a couple of examples of it. So we can culture cells without serum. So this is, on the y-axis is number of cells. On the x-axis is a number of conditions where we culture the cells. GM is with serum. You see the cells grow very, very well. It's after 1, 4, and 6 days. But we also have a couple of feed systems for these cells where we don't need the serum. And this is, of course, the way to go when we are still kind of optimizing this and making this cheaper, because cost, of course, is also an issue. It also needs to be efficient, meaning that we have to be more efficient than that cow, because eventually this is also a system that converts plant-based materials into animal proteins, very similar to any animal. So it only makes sense to do that if it becomes more efficient. Now, the advantage here is very similar to the greenhouses that Larry showed, is that it's a contained system. So you can change the conditions, you can optimize the conditions, and you can also start to recycle things. So in order to scale up production, cell culture, these cells have to adhere to a surface. So that's why we usually culture them on Petri dishes. And if you want to go to a large tank, you need to train them to grow on other surfaces. So we use little spheres that are kind of submerged, suspended in a big tank with fluids, and all the white dots on these spheres are cells, and they grow very well on it. And this way you can scale up to, commercially these tanks are now like 25,000 liters. That feeds about 10,000 people for a year, at least 10,000 Europeans, about 5,000 Americans. Still a lot, right? So we can culture them, we can scale it up, no problem. If you think about, so this is my exponential curve. It looks very linear, right? But that's because I used a trick, I made the y-axis exponential. So it's transferred into a linear curve. And if you do that math, if you start with one cell and that cell doubles 25 times, it's like that checkboard rice story, right? And if you do the math from one cell, you can actually create, at the moment we are now at about 25 doublings, you can create 35 kilos, 34 kilos of beef. That's a magnification factor of about 15,000. That means effectively that you can, 100,000. That means effectively that you can reduce the 1.5 billion cows that we have on this planet to 15,000. And actually we're currently at 35 doublings, and since it's exponential, that creates like 8,000 kilos of meat from one sample. So that's the power of this multiplication factor, and these stem cells basically, because they do all that work. When we do cell culture, and a lot of my colleagues in the medical field usually criticize me for that. Well, say, well, cell culture is not very efficient. We know that we have been doing that for a while. It's very expensive, not very efficient. One of the reasons why it's not efficient is because after 3 days, we throw away the food and we add new food. The reason why we're doing that is because these cells are basically growing, they metabolize and they produce ammonia as part of the metabolism. And typically, if this happens in people, we kind of pee ammonium out of and excrete that from our body. But these cells don't have a urinary system and they don't have kidneys, so they are basically growing in their own pee, if you like. So we need to refresh that, but every time we refresh that, we lose a lot of nutrients that are still in there. So we have analyzed how many nutrients are actually still in that system when we culture the cells for 7 days and we throw it away. And it turns out that about 50% of all the nutrients are still in there. So if we just remove the ammonia, which we're currently doing, which can be used as fertilizer and things like that, we can actually make that culture medium last much longer. And this is also the cost factor. Culture medium is by far the biggest cost factor in making cultured meat. By the way, for regular meat, the feed of the animals is exactly the same. That's the biggest cost driver. Of course, we also need to mimic meat as much as possible. You've seen the Impossible Burger, and they tried to do exactly the same. It's a plant-based burger, but they kind of introduced a hemoglobin type of molecule so that it looks like it bleeds. And so from a appearance point of view, it mimics meat. And they claim that also, this is, by the way, kind of controversial, but they claim that heme iron is also a big driver of the taste. That's somewhat controversial. But also the texture is very important. Meat is a very special product that we really like. By the way, who of you is vegetarian? Right. I expected a little bit more, to be honest. But it does illustrate that probably, let me ask, I usually do this in the reverse way, but who of you knew already of all kind of the problems with meat production, the environmental issues? Quite a few more. Right. You are apparently a very highly educated crowd. Sorry? Right, okay. Yeah, well, that too. Well, who of you knew it before Larry mentioned it? Still the same, yeah. Lowry, you didn't mention anything new, I guess. At least not in this in this area. All right. So that illustrates that sort of the rationality of the whole thing doesn't translate into human behavior very easily, right? And if anybody had to be vegetarian, it should have been me, but I'm not vegetarian either. All right, so that hamburger in 2013 was dry. And the tasters quite honestly mentioned that it's a little bit dry because there was no fat tissue. In the medical field, there are not a lot of reasons to produce fat tissue. So we had to kind of reinvent that. And we did, and now we can very effectively, with natural systems, produce fat tissue. In the next version, somewhere next year, we will have the muscle complemented with fat. And I will come back to that a little bit later. Once you start thinking about this, why would you stay with a hamburger? Why not, use exotic animals or mixtures of animals? Not by genetic modification, but by just mixing cells of creatures. Kind of interesting, kind of cute, and it might happen. What I find more important is that we can actually coerce these fat cells that we're making to produce more omega-3 fatty acids, so that the hamburger actually becomes healthier for you and starts to reduce your cholesterol instead of increasing your cholesterol. That's our gift to McDonald's. And that definitely is possible. So we can start to tamper a bit with it. Again, not with genetic modification, but just with picking the right cells and feeding them the right thing. So we can do this with beef, and I think beef is an important target. Was kind of coincidental because Sergey Brin, who funded this, said you have to make a hamburger. I wanted to make a sausage, but I said no, hamburger. So we ended up doing beef, and retrospectively I found that a very smart choice because those cows are by far the least efficient of all the animals that we have out there. Pigs are more efficient, chicken are even more efficient, fish is almost similar to a plant. However, there are other companies that work on chicken, super meat in Israel, mostly because of animal welfare, because of course there are a lot of animals involved in that industry. And friends of mine in California are working on tuna because of biodiversity reasons. Every creature that has these stem cells in their muscle, and as far as we know, mammals, fish, birds have that, you can do this with. And in fact, it is being done. Up to, this is a very busy slide, and it's meant to impress you, and I hope it does. Up to 3 years ago, I was the only one in the world, or my lab was the only one in the world doing this. And after 5, 6 years, it feels like a little bit awkward that nobody is following you. You start to think, am I an idiot, or is nobody else understanding this? And one of the most gratifying things, I must say, of this whole kind of endeavor is that since 3 years, a lot of companies are popping up. And it's almost like an exponential growth in and by itself of startups that one way or the other are involved in this. It's on the upper left corner are the protein producers, the alternative meat protein producers are like 35 of them. I need to update this slide every couple of weeks. Like 35 of them, chicken, fish, meat, pork, duck, but also milk, as Lowry already mentioned, for a perfect day. And what's more, and it's kind of difficult to appreciate from this slide, what's more is that a lot of investments are starting to flow into this industry. Not only from animal welfare people, also from animal welfare people, but also from larger meat industries like Tyson and Cargill and in Switzerland, Bell, who are investing in these endeavors. And in my conversations with those people from the meat industry, you really get the feeling that they realize that there is a problem coming up with meat production because we cannot really supply the increasing demand in the world, and that they don't really have a good solution for this either. And they want to become part of this and want to start thinking about this and also, to be honest, hedge their risk. Also, venture capital is now investing in it, so there people start to think, oh, wait, there might be money in this, right? I told you about the chloroplast that invaded our plant cells 800 million to 3 billion years ago. What if we can actually get chloroplasts in these muscle fibers so that they create their own energy source? Right? You will have green muscle, obviously. So it has a whole new, I don't know who of you grew up with Dr. Seuss, green eggs and ham. This would be something like that. It would create its own energy source. You have to expose it to sunlight. This might sound like science fiction, but trust me, it's being done in labs at the moment, successfully done. You can transplant these chloroplasts sustainably into muscle fibers. And yes, they do exactly what you expect them to do. This is going to be very similar to genetic modification, so it's maybe a little bit iffy, but yeah, it's going to happen. We make hamburgers, and Laurie, I guess, mentioned in a justifiable way that hamburgers can be kind of mimicked with plant-based. I'm not totally convinced, but they can get a long way. But how about ribeye steaks, which would be my next sort of challenge? And this medical tissue engineering technology can get you there without too much problem. We're starting to work on that. When we presented this in London, the popular press said, yuck. And I was kind of frustrated by this yuck because I didn't quite understand what was behind it, sort of an emotional aversion against the technology. And my dilemma was this, it's a hot dog. Who of you has ever eaten a hot dog? Do you know what's in it? Do you want to know? No. We are perfectly capable of eating stuff that we don't know what's in it or how it's being made. So how's that different for my hamburger? And my kind of thinking about this, a lot of this has to do with safety. We see our neighbor eating hamburgers, he stays alive, and we say, oh, come on, it's probably okay, we can try it. So here, for every new product, we are kind of biologically programmed not to eat stuff that we don't know. If you go into the forest and you start picking mushrooms and you don't know what you're doing and you put them in the soup, you have a big chance that you poison yourself. So very useful reaction. So we need early adopters, and we have them enough, and then obviously they need to stay alive also, and then it will take time. That's it. I'm not too worried about that, to be honest. The other thing is that we want to have the illusion of control over how our food is being produced. And that's a lot with other technologies, where we see the technology and we kind of think how this is going to be implemented in society, and we get worried. And here's the same thing. It's a cow, on the one hand, in nature, lots of checks and balances by nature. People can mess with it, but not too much, and that's it. Now you convert that into something that's completely made by humans. People can be fraudulent, they can make mistakes, they can be money-driven, and God knows what happens. So we developed kind of the story of a petting zoo in the middle of a city, let's say Helsinki. You have a petting zoo, 3 animals, three chickens, 3 cows, 3 pigs. Children of primary schools go there, they pet them, they give them names, they feed them, hence the name petting zoo. And in effect, they are stem cell donors. So once in a while, you poke them in the butt, and in a shed next to the farm, you grow meat for the entire neighborhood, like a microbrewery type of system. That would make it much more acceptable and much more controlled. So we did a study in the Netherlands where on acceptance, this was my first psychological study ever, it was kind of exciting. And we wanted to know what type of information actually drives this acceptance. So we split a general population into 3 groups. One got information about personal benefits, one got information about societal benefits, environment and stuff like that, and one got information about this is really good meat. And then we did something unusual. We had them taste a piece of conventional meat, labeled as conventional meat, and a piece that was labeled as cultured meat. They were actually both from the same hamburger because it was a psychological study. So here's the results, kind of interesting. So the colors are the benefits, so societal benefits, personal benefits, and quality. We asked the questions before we gave the information, after the information, and after the tasting. And we asked, are you in favor of this technology? Do you want to taste it? Do you want to buy it? And do you want to replace your current meat consumption with cultured meat? And what kind of interesting was, and you see that immediately, although it's a busy slide, you see that the colors actually don't matter. So whatever type of information you give doesn't really matter in acceptance. You also see that every type of information that you give increases acceptance. People are really information hungry if it's about a product that is new. What was also interesting that almost 90%, or even more than 90%, actually wanted to taste the hamburger. We are kind of curious. And we realize it doesn't kill us immediately. So the safety issue is not really sort of the immediacy thing, but a long-term thing. What was interesting is that everybody ate that piece of meat that was labeled cultured, no problem, because of course it looked nice and it looked like a regular thing. And they found that one better tasting than the other one, although they were exactly the same. Because if you tell people it's very special, it starts to taste better. That's just what we are pretty gullible in that respect. So my vision for the future is actually very, very simple and romantic, in spite of all the technology that we're working on. It's continuing to use meals as sort of an excuse for social interaction in a very nice atmosphere with friends and family. Being able to eat all those wonderful things, including meat, that we have gotten used to and we will increasingly get used to, without the negative consequences for the planet, for the animals, and for food security. Thank you very much.